Mira. No, it's my mom, but whatever. Who's it? I don't know. No tengo pruebas, pero tampoco tengo dudas.
Good morning, everybody. So how is the beginning of the semester going along? 3 weeks are over, right? So you are now into it, full block. Yep. Good. And so senior design is around the corner. I can't believe it. So its stress level went up?
Yeah.
No. We have an anesthesiologist speaking, so maybe. Yeah. So it's my great pleasure to welcome Dr. Carlos Mantilla today as our speaker. We were just talking that when did I first get to know Dr. Mantilla? Probably we're thinking at least a few dozen years ago. And so it's been a long time that we have known each other. But what, and especially the grad students who are here to understand, is that those national conferences you go to, those become kind of like a friendship conference place. So over the years, I think we regularly meet every year at those conferences where we are going to see his posters, he's coming to see our courses. He was just asking about Ricardo Su. Many of you grad students know Ricardo, he just graduated. And those of undergrads know because he worked with you all a lot. Like, where is Ricardo? Well, because he's been watching where is Ricardo all the years as Ricardo was growing through his grad school years, right? And Ricardo, by the way, is at University of Louisville doing research with human subjects already. He's already into it, and he just sent me an email how excited he is about that. So anyway, I've known Dr. Mantilla for several years, and what is neat about him is that he has got this very broad experience. So he has got everything that we try to teach you all, and he may be an anesthesiology chair, but I think he's a biomedical engineering chair, which he is the chair for the director for graduate studies. But he does computational modeling, he does basic science, molecular level research, and he does human subjects. He works with people, right? So he's an MD PhD. So those of you who might be considering the both ends of the world, talk to him if you are at lunch, grill him about that, or what is that path like. So he has been able to bring all of those together. I know several of you have family roots in Colombia, and that's where his roots are from. So it brings, right? So that is another link that we have with Dr. Mantilla, and that is another thing that you can talk to him about as to what was the transition like? What does it mean to have come from one place to another and move through the whole structure there? He's at the Mayo in Minnesota. Minnesota has a fantastic ecosystem for entrepreneurship and especially medical device industry. And so Dr. Mantilla is engaged with that too, and he also can give you information on that. The topic today is going to be about the respiratory system, diaphragm muscle function, and I will let him talk about it more. And earlier he had a slide up there about a summer undergraduate research fellowship program, a SURF program. So undergrads who want to do internship also have opportunities to go to Mayo. And Mayo, as you all know, is a world-class premier both research, clinical institution. So welcome.
Thank you for the very kind words. Just checking, you can hear me in the back okay? There's seats in the front, so no microphones up. Not working so well?
Not working, so is it on?
Yeah. Let's test it again. You can hear me better now? Okay, very good. So thank you very much for those very kind words. I now wish I had recorded them to share them with my family. Missed on that chance. So I want this to be very informal in the sense that please interrupt me if there's any questions. Just a sense of who's in the room. How many of you are undergrads in biomedical engineering? Okay, how many are graduate students in biomedical engineering? How many are faculty in biomedical engineering? Okay, and how many are in other engineering fields that happen to be here? Okay, very good. So I have had the privilege of working at Mayo for a number of years, and as Remy was saying, I'm both a physician, anesthesiologist, as well as a researcher, and I interact a lot with many of our colleagues in biomedical engineering and physiology, and I have the privilege of being the graduate program director. So the chair for the Department of Biomedical Engineering is Wyatt Prakash, and I think many of you might also know him, also in respiratory. So we have a very strong group of folks that work in respiratory systems. I understand that there's someone watching on video somewhere, so I'll try not to stand in the way of the slides as I'm going through. So, welcome. So today I really wanted to touch three main things. One, how is motor control of the respiratory system implemented? Because many of us don't really think about breathing, but yet it's one of those things that you have to do the very first moment you're born, and you cannot stop doing for the entirety of your life. So as an anesthesiologist, of course, I think that breathing is one of the most important functions. Right? Without breathing, you won't survive the surgery. So we, of course, have a number of interventions for that, but it's something that I think we still don't understand from the central nervous system, how that control is implemented. The other parts that I wanted to talk to you was give some case examples of what happens across the lifespan and the change and what changes occur in the respiratory system and the control of breathing, and include some work, some of our work in spinal cord injury. And lastly, I wanted to touch on the clinical aspect of this and how we could take this to patients. So again, please go ahead and interrupt me if there's any questions. So I want to start first by thanking the folks in the lab. This is our regenerative physiology laboratory. We have a number of folks in there that might be familiar to those of you that go to experimental biology or Society for Neuroscience. They've been presenting our work there. And the work that I will show you today is primarily Savia Rana, graduate student in our program, Maria Gonzalez, another graduate student in our program, Heather Grancy, a research associate in our program, and then Richie Jean and Gary C. that have been longstanding collaborators with me. I also want to thank our funding sources. We've been very fortunate to have been able to be funded for a number of years, and of course the Graduate School of Biomedical Sciences, because they allow us that contact with the students and faculty. So what is the diaphragm muscle? So many of you know this from biology in high school, but the diaphragm muscle spans the abdominal and thoracic cavities, right? It's a barrier between the two of them. That's where the name comes from. But it is also the main inspiratory muscle. So in humans, this is the muscle that you use for breathing. Most mammals, it is the case, including quadrupeds. It's a very active muscle, as I said, you use all your life, but not only is it active all your life, it's active a large proportion of the time. So when you think of other muscles that are active, like that you use for standing, leg muscles that are used for standing are active somewhere between 5 and 12% of the time. The diaphragm is active up to a third of the time. So a very, very different levels of activation. It's also not gravitationally loaded. It's only loaded by the respiratory system mechanics. So when our astronauts and pilots go into outer space, and maybe tourists in the near future go into outer space, the diaphragm won't atrophy, right? But all their other limb muscles and so on will, because they are gravitationally loaded. But very importantly, in terms of understanding motor control in general, it represents all types of motor units. So you might be familiar with the concept that there are some muscles that are really strong and don't tire out, and other muscles that are not, that are much stronger, but that really tire out. And there is a balance amongst those. The diaphragm has all of those represented. So I want to, even though I start with breathing control, I also want to remind you at this time that the diaphragm does many other things beyond breathing. And that's what allows us, again, to use it as a model to study many other things. So what is a motor unit? Sir Charles Sherrington, who was awarded the Nobel Prize in 1906 for his work as motor units more than 100 years ago, coined it to essentially represent the motor neuron that is shown there by that stellar body over here, and all the muscle fibers that it innervates. So that is a motor unit. After development, motor units are organized such that one motor neuron innervates a number of muscle fibers down here, but each one of these muscle fibers is only innervated by one motor neuron, right? So that's not the case early on during development, but fairly soon after we're born, that gets established. We're not going to talk much more about that unless there's questions, but that principle of organization is very important because the properties of the motor neuron and all the muscle fibers that are within that same motor unit are shared. So even though there are some weak and some strong, some fatigable and some fatigue resistant, the properties within a motor unit are common. So this is exactly to illustrate that point. There are what we call type S motor neurons because they're slow twitch. They generate very low levels of force, and I will continue to apologize and just reflect the historical significance of using grams as a unit of force. All of you engineers will know that dates this type of slide many, many years ago. But that is just to show in the height of the bar is the force elicited by one motor unit of that type. So as you can see, type S motor units generate much lower levels of force than type 2X motor units. But type 2X motor units will drop their level of force with repeated activation, in this case over 2 minutes, whereas type S motor units and even type FR, fast twitch but fatigue resistant motor units, don't lose that force. So which motor units are active is very important for us to understand because it will determine the complement of behaviors that the muscle can accomplish. This applies to every muscle, not just the diaphragm muscle. There's a general organizing principle of how the central nervous system controls motor systems. So in the diaphragm muscle, there's a number of ways in which you can measure the forces and the activity of the muscle. So at the very top, you have electromyography. So how many of you use electromyography for any application? So a few. So in this case, we have on the left column says resting breathing, also called eupnea. And the little twitches, the little twitches over here are electrocardiographic interference, right? You just get a little bit of artifact from the electrocardiogram. When the animals are exposed to hypoxia hypercapnia, that is low levels of oxygen, high levels of CO2, the respiratory drive gets increased, and then they breathe a bit faster and stronger. As you can see here, the amplitude seems to be a little bit more. And then when you occlude the airway so that the animals are trying to overcome that resistance to breathing, they breathe even faster, harder. But it is not until you use airway clearance behaviors, like sneezing or coughing, that you actually get into the higher levels of force. So that is shown over here. So I won't really have time today to show you that we did a validation and a comparison of various methods of actually quantifying the electrical activity of the diaphragm. And the root mean square, the RMS, is the one that associates with the diaphragm activity regardless of position, regardless of muscle shape, regardless of lung inflation. The best. Because you can also measure electrical activity like this by using rectified integrated values. You could use something like fixed sample entropy to look at these samples to get additional measurements and eliminate artifactual information like the electrocardiogram in one swell soup. But what it doesn't do is correlate as well with the transdiaphragmatic pressure. So one of our engineering students did this validation studies for looking at what is the best measure for quantifying EMG and its relationship to the forces that the diaphragm develops. And in this case, we use transdiaphragmatic pressure, or PDI, the differential between pressures in the abdomen and the chest cavity as a measure of force. So as you can see here, the force elicited by the diaphragm goes from small during resting breathing, more during hypoxic hypercapnic exposure, even more during airway occlusion or sneezing. Okay, so the highest levels of force that the diaphragm elicits, even in humans, this is rats, but even in humans are really not during breathing, not even during exercise. When you think you're breathing hard, that's still not the maximum force. It's really when you cough or bear down, and you would say, but coughing is an expiratory, sneezing is an expiratory behavior, right? The air's coming out of the lung, not going into the lung. Well, it turns out that it actually is multi-phased behavior, right? You need to have a big lung capacity to have a lot of lung air volume to expel what is in your throat and cough effectively. So the very first phase is actually a large inspiration that is driven by the diaphragm, followed by active contraction of the diaphragm so that that allows greater pressure generation. So I took a long time to explain this because then I'm going to go way faster through all the various models. So this is a model that Gary Seek developed many years ago now, where we essentially looked at what was the maximum force that a motor unit of each type, so the type S motor unit generates this level of force compared to type FF, and then looked at the proportions of motor units of each type, and we've done this for dogs, cats, humans, mouse, rats, turtles, and piglets, right? So across many different species, you can generate models like this. And you use the slope as the force per motor unit, the proportion of motor units, to get at the total levels of force on the y-axis, and then correlate the behaviors as they compare to the maximum. As I said, respiratory or ventilatory behaviors in like eupnea, hypoxia, and capnia only elicit a fraction of the maximum forces that are developed and can be fully accomplished by full activation of the fatigue-resistant units. That makes perfect sense, right? Why would you use a unit that is fatigable to do something that you need to do all the time? You want to have units that don't tire out to do those types of behaviors. So for the diaphragm muscle across species, those fatigue-resistant units, type S and type FR, are sufficient for us to be able to elicit ventilatory behaviors. So our work for a number of years has really delved into this. So how do these interactions between the motor neuron and the muscle fiber lead to the properties that are homogeneous within a motor unit, but yet heterogeneous across motor units in the same motor? So that could it be genetic factors? Could it be the activity, right? Diaphragm is a very active muscle compared to other muscles. Is it neural-derived influences, muscle-derived influences? Those are things that we will start dissecting throughout the talk and in our work. And of course, there's more work to do. So we, a number of years ago, developed a full characterization of what was available in the literature about motor units and trophic factors, so known factors that are or have been described as exerting trophic growth influences on motor units. And the ones that I'm going to focus on today is this one, the brain-derived neurotrophic factor family that activates a full-length TrkB receptor, tropomyosin-related kinase, that's a tyrosine kinase, and that receptor. So that's really where most of the trophic factor work that I'm going to talk about. I'm going to use this type of representation as a way in which we can measure the efficacy of neuromuscular transmission. So in this particular case, we have a setup where we stimulate the nerve, and we do that repeatedly once per second. This is in a dish. We have muscle within force transducers, the nerve still attached and intact, and we can stimulate either the muscle directly or the nerve directly. So measure the force as a measure of, as a fraction of the total force elicited by the muscle, how much of it can be elicited by the nerve. So with nerve stimulation, as you can see there, it starts pretty close to the twitch with muscle, somewhere around 95 or even 100%, and then it quickly declines as that nerve becomes ineffective at eliciting more muscle contraction. Even though when the muscle is stimulated directly, intermittently every 15 seconds this time, you can see that the muscle itself hasn't fatigued, right? So the difference between the forces elicited by the muscles and the nerve down here, that gives us an index of neuromuscular transmission. How effectively did the nerve elicit muscle contraction? So this is a summary slide of many such measurements. And you can see the muscle stimulation stays pretty high, levels of force, nerve stimulation declines. But when we give something like BDNF or another TrkB analog, neurotrophin 4, both of those reduce the difference between the nerve and the muscle. And remember, because the muscle is fatiguing, you can't take them in isolation. You need to consider them together. So that to us suggested that neuromuscular transmission was improved. And then conversely, if we use a tyrosine kinase inhibitor like K252A, or a fusion protein that has essentially the binding domain of the TrkB receptor, but makes it soluble so it quenches any available neurotrophins, that had similar levels of worsening of neuromuscular transmission. Again, to show that the BDNF and or NT4 are playing a regular role in controlling the level of neuromuscular transmission. We then took advantage of a genetic mouse that was developed by David Ginty at Johns Hopkins. And this is a mouse that has a single point mutation. Substitution of F for A, that amino acid, that's in the ATP binding domain of the kinase for TrkB. And that mutation makes the receptor work perfectly normally. So these animals develop as any other animal. But when we give them a PP1 derivative, protein phosphatase 1 derivative, like 1-NMPP1, you can block the activity of the kinase. So that allows us to selectively, rapidly, reversibly inhibit the TrkB kinase wherever we want. So with this mouse, we first validated that the neuromuscular transmission failure in the untreated mouse, here instead of showing both muscle and nerve, I'm presenting now the index. So neuromuscular transmission failure was not affected by 1-NMPP1 treatment in a normal mouse because they don't have the mutation. Neuromuscular transmission failure recovered with no 1-NMPP1 and was normal. But with 1-NMPP1 in the light blue, the neuromuscular transmission failure was worsened. And so neuromuscular transmission is impaired when TrkB kinase. So we went on to do a number of studies characterizing what happens over time, and it is quite well established now that TrkB activity is essential for the regulation of the size, the shape, and the function of neuromuscular junction. So this here is a label for presynaptic terminals. That's synaptophysin on the left in green. Bungarotoxin in magenta shows a postsynaptic end. So you know the neuromuscular junction is the place where the nerve meets the muscle. So there is specialization, and that's called the NMJ or neuromuscular junction. And then you can see that with 1-NMPP1 for 1 week, they become abnormal. But when we restore TrkB function, they have recovered. So that's in terms of the morphology. We can do this in 3D using some fairly customized algorithms we developed for image processing that allow us to do that at high throughput. So we can measure thousands of neuromuscular junctions in a single muscle from single application. And you can see that presynaptic volume changes with just 1 week of treatment and 1 week of recovery, and motor end plate volumes also change. So in vivo, we can now demonstrate that these things that we have tested in the dish have long-term implications for plasticity of the neuromuscular. And the other point that I think is important to reflect on is that the central nervous system, and in this case peripheral nervous system, are highly plastic, right? Even though we think of our brains as being matured maybe at age 8 or something like that. Maybe you will debate with me at what age that happens, that's okay. But after our brains mature, there's still a lot of room for things to recover and improve, and that can be harnessed then to have our patients experience recovery from disease. So Maria worked on then trying to deliver nanoparticles to the motor units in a selective way. So I won't show you her work here, only to say that for that we had to understand how is the trafficking at the neuromuscular junction established. So she worked by understanding lipid raft uptake. So lipid rafts are domains within the cell membranes that agglomerate proteins that have shared function and allow them to be tightly put together so that, you know, neuromuscular transmission can be effective, for instance. So this is looking at, again, synaptophysin on the left, at different frequencies of stimulation in the dish, and then colorotoxin B as a marker of the uptake. Why? Because the protocells, the liposome encapsulated mesoporous silicon nanoparticles that we developed were functionalized by adding colorotoxin B so that they would be taken up by motor neurons. So you can see then here just from the images that there seems to be more black at higher frequencies, both in the smaller type 1 or 2A fibers corresponding to the type S and type FR, fatigue-resistant motor units, compared to the type 2X and or 2B fibers corresponding to the more fatigable but fast-twitch motor units. So this is the quantification of many such motor units in animals, and you can see that at the higher levels of stimulation at 80 hertz, so from 0 to 20 to 80 in both types of motor units, there's a significant increase in fiber uptake, lipid graft uptake, even though there's quite a bit of variability across animals and even within neuromuscular junctions. So far, I think I've told you about our work that neurotrophins play an important role in neuromuscular transmission, that role is mediated by activation of this receptor, the TrkB kinase receptor, that it likely reflects changes in lipid graft trafficking. I didn't show you data to that, but that's actually ongoing work with Kevin Webb, a new grad student in the lab. And lastly, that stimulation frequencies might reveal that there are differences across motor units that are mediated this way. Why? Because the most fatigable motor units, the strongest motor units, actually fire at the highest frequencies of activation, whereas the fatigue-resistant ones usually fire at the lower frequencies of activation. So this would suggest that, especially in disease conditions, the most fatigable motor units are most vulnerable to any disruption of the neurotrophic signaling. Right? They depend on them more because that's where the firing can occur for them. So I'm going to take a slight detour and talk about spinal cord injury. So any of you work on spinal cord injury and rehabilitation of some work in some way, restoring function in different ways? You will agree with me that spinal cord injuries, fortunately, are not that common, but they are truly devastating. Right? And I would say 17 or just under 18,000 new cases a year is still very common. Right? One case is too many in my view. Overall, in the States, there's about just under 300,000 folks alive with this. And for the purposes of this talk, I want to remind all of you that what people die when they have spinal cord injury from is inability to breathe and respiratory complications. That is the main cause of death. In fact, the biggest predictor of how long a person will live after spinal cord injury is whether they're mechanical ventilator dependent at 1 month post-injury. So if they're on a mechanical ventilator 1 month after injury, their lifespan is going to be halved. Why? Because they're going to be subject to repeated infections, respiratory complications, and so on. So we believe that if we're going to give anyone a chance to use robots, neural stimulation, other transplantation devices, tissue-engineered devices to restore the function in other ways, we need to keep them alive and well so that they can do that. Addressing the respiratory system is one of the main things to do to make that possible. Obviously, it's very costly. And for those that are not in spinal cord injury field, the most common site of injury nowadays is the cervical spine. So incomplete levels of tetraplegia are now the most common. So we've used a model that, in fact, Coulter first developed in 1895. You can now say 2 centuries ago, but not 200 years ago yet. But it's a model that's been very well established across multiple species, including humans, where if you just section half of the cord, the right or the left side of the cord, you interrupt the descending inputs on this to the phrenic motor neurons on one side, and that paralyzes the side. As you can see here, the EMG recordings at the bottom, 3 days after injury, on the site of injury, there's no rhythmic activity corresponding to the breathing activity. But also, because it's an incomplete level of model of injury, there is spontaneous recovery over time. So it's not full recovery, but there is some level of plasticity that was evident even in 1895 that suggests that the nervous system, again, is recruiting those motor units on the injured side some different way from before. So that is what we've used to study the roles of neurotrophins in this context. So this is from a paper that we published a few years ago showing that when we give intrathecal into the cerebrospinal fluid, we can give both brain-derived neurotrophic factor, BDNF, or this fusion protein, TrkBFC, so that it quenches all the available BDNF. And when we do that, and we record EMGs here, you can see that 14 days post-injury in the untreated animal, this is one that showed recovery. The activity is very, very small. If you look in the middle graphs to an animal that received BDNF, that looks much closer to what it was before the injury. But if you look at an animal that received TrkBFC, the fusion protein, it does not recover. You can completely block the recovery. If you look further into each one of the bursts of EMG activity, you can see that there's what we call multi-unit So there's multiple motor units being activated, both in the untreated but normal condition in BDNF before injury, and also in the 14-day BDNF after injury. So this is a summary of many of these such studies. So in the white open circles, we have the fraction of rats that show any type of activity, any. And by us, any is, it had to be multi-unit, it had to be rhythmic, present 90% of the rats, and it had to be at least 10% of pre-injury levels. So these are chronically instrumented animals. It allows us to compare the EMG amplitude before the injury to various times after the injury. So in the open circles, you can see that roughly 40% by 14 days post-injury have some level of recovery. With BDNF, nearly all animals have some level of recovery. And with TREC-BFC, the fusion protein that takes away all the BDNF, no animals recover. Right? And these are now large series because we've been building on this since the very first observation. This is, again, very exciting. So what is it about BDNF that can help us enhance the normal plasticity that occurs post-injury, and how can we use that therapeutically for our patients' benefit? So we pursued this various different ways. I mentioned a little bit about the nanoparticle work that Maria mentioned, but there's other ways in which you can deliver drugs, right? You can do that intraspinal injection, like we were suggesting perhaps with the nanoparticles. You can deliver genes with cells engineering in vitro also to do intraspinal transplantation. All of them around the area of the phrenic motor neurons to try to elicit plasticity. You can also do remote gene delivery. And one of the ways in which we pursued that was realizing that the nerves that go to the diaphragm muscle are in a cavity, in the chest cavity. And that cavity has a lining that is called a pleural lining. That pleural lining has a little bit of fluid that surrounds all the neuromuscular junctions. And in the clinic, we use this to deliver pain-relieving medications for our patients that have chest surgery. So a number of years ago, well, I thought, well, why can't we use that to deliver things directly to the motor neurons without having to do intravenous injections and so on? And we were fortunate enough that it does work. So we've demonstrated this, and many labs have since taken this approach, where in this case we used intrapleural toxin B, and also labeled all motor neurons with cutting the nerve, which was the old way of retrograde labeling, right? You wanted to see how the nerve worked, but you cut it so that you could label the motor neurons that went to that nerve, kind of defeating the purpose in many ways of your experiments. So in this case, we use rhotamine dextran in red, and you can compare here that they very closely match. In fact, we've shown that more than 95% of all motor neurons on the side of the intrapleural injection get labeled. So, and no other motor neurons get labeled in the same levels where the motor neurons are located, phrenic motor neurons are located, which in rats is between C3 and C6, the third to sixth segment of the cervical spinal cord. But of course, this is in the chest cavity. So thoracic motor neurons that go to the intercostal motor muscles also get labeled, but they're in the thoracic part of the spine. So in the cervical spinal part of the spine, they're pretty specific, but they don't just label phrenic motor neurons. So this is one such label, that labeled motor neuron, so you can get very detailed labels of motor neuron that we can then do three-dimensional reconstructions and very sophisticated quantitative analyses on. We'll get to that in this talk. But we can also use it to then deliver viruses that we engineer to deliver specific constructs. And guess what? We went after the TREC full-length construct. So this is to show, we did a screen with at the time graduate student, but now Dr. Heather Grancy, looking at what types of serotypes would get to motor neurons in the most specific and selective way. Right, so we didn't care to label all the motor neurons because we could study one motor neuron at a time, but we wanted to make sure that only motor neurons were labeled so that whenever we did something, it wasn't because of complex intracellular communication and other plasticity, it was because it happened at the motor neuron, nowhere else. So that's what we value most, and as you can see here, we have retrogradely labeled phrenic motor neurons in red with cholera toxin B, so all of them get labeled. You see big bunches of motor neurons like that. And when we use AAV2/7, which is for human application, you can get, in this case, green fluorescent protein in one of those motor neurons. And you can see it in the vertex grayscale, so it looks a little bit easier to see. So this construct had flag, and you can see here the entire cervical spinal cord homogenate, we can detect the flag protein. So again, suggesting that even though we're only targeting a fraction of the motor neurons and only a fraction of all the cells in the cervical spinal cord, the expression was robust enough that we could detect it by common measures of protein expression. And that's what is shown. So what happens when we deliver intrapleurally AAV that delivers TrkB for the frank motor neurons? Well, we get 100% of rats displaying some level of activity by 2 weeks post-injury. And we get roughly 75% of the amplitude of the activity from before. So think about this. Somewhere between 10 and 50% of the 300 or so motor neurons on one side became transduced with this virus. No other cell was manipulated in any other way, right? But that's sufficient for us to restore breathing, right? Obviously, it probably won't be sufficient for us to restore cough, right? Because it's only 10 or 50% of neurons, but sufficient to restore breathing, right? Why is that happening? I'll show you a little bit of what we think, how that's going on, but won't get into that a lot. We did the converse experiment. We now use an siRNA for TrkB, delivered only to frank motor neurons, and we can abolish. There we go. And again, showing that TrkB overexpression good, TrkB knockdown bad. If we think recovery is good or bad, right? It's probably good. Then we did this waiting some time after injury to see if giving the virus was sufficient to recover after injury. So Gabriel Martinez is a graduate student at the time, or is a graduate student in the program at this time. And we did, again, 3 days post-injury, now we give the virus. So the previous one was a proof of concept. We had already overexpressed or inhibited the TrkB before the injury. Here we gave it afterwards. And to make this story short, we get good results, not quite as good as in animals that have overexpressed TrkB levels. So only at 75% recovered, not 100. And the level of recovery is not quite as good, it's only about 40%, right? So promising, but obviously insufficient. So one of the ways in which you can approach this, there's multiple ways now, but at the time we pursued laser capture microdissection. It's a tool that allows you to, in frozen tissue that you have labeled some way, pick up the cells of interest for further analysis. And in this case, we use quantitative real-time RT-PCR or real-time RT-PCR to measure glutamatergic receptor expression. Why? Glutamate is the main excitatory neurotransmitter in the central nervous system, including at phrenic motor neurons. Right? So if you're going to drive phrenic motor neurons to be reactivated, glutamate is likely the neurotransmitter. So we looked at the receptors that are both ionotropic receptors are AMPA and NMDA. And if you compare the animals that received the virus in the red compared to just injured animals, they all moved higher levels of AMPA and NMDA. So these are individual animals on average, what their motor neurons do. If you look further at the NMDA levels in motor neurons from control animals, they're low. They get higher with just giving the virus. They get higher with injury, so obviously the plasticity that is occurring is eliciting some changes in NMDA receptor expression. And they get even higher with the TrkB receptor. So this is very promising because TrkB likely regulates NMDA receptor expression. So Sabia Rana, who is now at the University of Florida and finished her PhD just last year with us, did single-cell multiplex in situ hybridization experiments now. So instead of looking at one gene at a time or one cell at a time, we can now look at multiple genes all in one cell, compare cells instead of homogenates or aggregates of multiple cells picked up per cap. Now we have cell by cell. This allows us much more powerful. So if you see here in magenta, this is toxin B labeled motor neurons. We use that outline then to identify them and then measure. These are RNA transcripts for AMPA or for NMDA, and this is the merged image, right? So we can now in this same cell, just cell by cell, not aggregate per animal, look at what is their AMPA and NMDA receptor expression. So, and what happens with that after spinal hemisection and with TrkB. So I'm going to cut to a chase. And I'm going to tell you here, if you look in the pseudo color scale on the right, the blue colors are low levels of expression of motor neuron size. The red colors are larger motor neurons. I didn't mention this until now, but the size of the motor neurons actually matches some of their physiological properties. The smaller motor neurons are associated with the fatigue-resistant units. The larger motor neurons are associated with fatigable, but stronger, fast-twitch units. It's not a one-to-one relationship. There's a lot of shades of gray in that spectrum, but it kind of generally holds. So in this case, we're again plotting the TrkB receptor mRNA versus NR1, one of the subunits that is mandatory for all NMDA receptors. And as you can see here, that 3 days after injury, most of them are clustered where there's low levels, and they're all together, regardless of motor neuron size. Maybe a little bit of the smaller ones, a little bit over to the end. But by 21 days post-injury, the smaller motor neurons in particular, the fatigue-resistant motor units now, because the blues are extending outwards, have higher levels of TrkB receptor expression and higher levels of NMDA receptor expression. Right? So these types of experiments are ongoing to really help us dissect what happens now when we give BDNF, what happens when we give the TrkB virus, what happens if we block TrkB synthesis. Can we change all these? Those experiments are ongoing. So I hope to have more information for you at the next meeting. So the bottom line is that increased ipsilateral expression of both TrkB and NMDA occurs primarily at those smaller phrenic motor neurons that we think reflect the fatigue-resistant motor neurons. So what happens now if we pharmacologically go old school, and just apply an NMDA receptor antagonist, so just a dirty drug, onto these motor neurons, and just giving an NMDA receptor antagonist 28 days post-injury, an animal that had recovery, the animals, you can block recovery. And suggesting to us that that TrkB enhanced recovery is mediated by glutamate, right? So it's kind of circling back on the story that it is indeed the receptor. So I've told you then that restoration of this rhythmic diaphragm activity reflects neuroplasticity in this model of incomplete injury, but that it doesn't activate all motor units, so we still need to work on that. But targeting gene delivery to phrenic motor neurons is sufficient to enhance recovery. And improving the ability to generate these higher forces will be necessary, and we have not been as successful with that yet. I'm going to go now and switch gears and talk to you about another condition where it's important to understand motor control, and that is aging. So similar to spinal cord injury, the person that is in a nursing home that is perhaps has dementia and that is aging, but their body is otherwise healthy, actually dies most commonly from pneumonia. In fact, the common word in geriatric circles is that pneumonia is the old person's friend, because it's a fairly peaceful way in which people seem to pass. But why then are we content with that? Why don't we try to do something to prevent respiratory complications at home? So we think that there's multiple things, of course, that are happening when we age, but the respiratory system mechanics are changing. The chest is getting stiffer, and that should increase the load. As I mentioned, the diaphragm is loaded by the respiratory system mechanics. There's also atrophy. As you know, as we age, we tend to have more difficulty maintaining muscle mass. And in fact, loss of muscle mass is one of the biggest predictors, regardless of species, of death within a certain number of months. So when any of us lose somewhere between 30 and 40% of our muscle mass, the likelihood of death within 6 months is increased 2 to 3 fold. So if you see that in your great-great-great-great-grandparents, that's usually a fairly good indicator that things are no longer working as well, and their resiliency is being compromised. Mentioned neuromuscular transmission failure at the very beginning. In aging, we're going to use that again to study what happens and what is the role of these trophic interactions. So we're going to focus on BDNF and TrkB mediating neuromuscular disruption that we think contributes to loss of muscle mass and force, also known as sarcopenia, that reduces the maximum transdiaphragmatic pressures that can be generated, such that the reserve capacity, the difference in the ability and the forces that are necessary just for ventilation, and the forces that can maximally generate it is reduced. Right? That reserve is being lost. That is what leads to ventilatory, non-ventilatory behaviors being impaired, inability to clear the airway, and the age-related respiratory complications. So we measured with John Elliott and Sarah Dreising, postdocs in the lab, airway mechanics, and as you can see, as rats age, their airway resistance in fact goes down, and airway compliance goes up, reflecting in mostly destruction of the lung parenchyma. So the lung units, the units that are necessary for gas exchange, instead of being small, now their walls get broken down and they become large. So their compliance and resistance go down, but that's not a good thing. It's a bad thing. The functional units in the lung are going down. And there's also something that happens when you look at the very, very old. And I won't have time to dwell into this anymore, but when we study the very old, we study the survivors, right? So we see this U-shaped across a number of conditions where things are getting worse as you age, and then suddenly the really old are not so bad. But it's because they made it. If they had had some of the other things that the rest of the population had, they wouldn't be that old, right? So it's something that when we look at aging, in any natural aging model, we always need to remember, right? Because you're studying survivors in the very old. In fact, we just published a paper with an undergrad student, in fact, that worked in our lab, Pandra Vaughn, and her summer project was to look at what happens in the really, really old ones. And the entire paper was to say they're actually better than the not so old ones, right? That was the entire conclusion of the paper. They suggesting that there is a floor. You lose function, and when you lose any more function, you just die, right? So anyone that you can study that is alive has at least this level of function, right? So again, those are things that I just want to mention because it can confound how we interpret some of the results and what comes up in the regular press all the time about this good for you, this bad for you. Well, it's because that's right mixed in there, right? Because if they're studying the athlete marathoner that is running their 100th marathon at age 95, they are not me, right? That's not what I will look like when I'm 95. So that's so. In old age, there is sarcopenia. So there's reduced force. These are stimulation frequency dependent changes in specific forces. You can see in the open circles, we have the male and female young. And in the gray circles and squares, we have the male and female old. If we look at this is across frequencies, as you can see, there's no real differences across sexes in rats at these ages. We've used an age that roughly 75% of animals have still survived, so only 25% mortality. That is one way in which we can compare ages, right? What age do people die? So as you know, in the U.S., we were having a declining survivorship. I think this year it's the first and fourth that the survivorship is increasing. The average life expectancy for folks in the U.S. is increasing again, which we would expect in every advanced civilization. So anyway, in the old animals, regardless of sex, you can see that the maximum forces are reduced compared to the young animals. If we look at these are now histological sections of the muscle. We've labeled the fatigue-resistant type 1 units and fatigue-resistant type 2A units in muscle fibers in red, and the black ones are unlabeled and are the larger 2X and 2B fibers. So we can use this, and we developed semi-quantitative processing in MATLAB. I saw that some of you use MATLAB out here to essentially automatically find the thresholds, find the centroid for each muscle fiber, detect the distances between muscle fibers, get us a mosaic of what the muscle fibers look like, and then give us also proportions and interfiber distances and all these other things to get at a sense of what happens with old age in these. So Obaid Karam, now at Northwestern University, but part of his PhD work was to essentially look at the old age effects. And you can see there's atrophy of fibers in old age, but only of the strongest 2X2B fibers. Right? So we talked about earlier that there could be vulnerability of these muscle fibers. We think that that vulnerability is expressed this way. They're more susceptible to the aging effect by showing atrophy. And so that suggests that the muscle atrophy that you're seeing is not just because something happens within the muscle, although that can of course be a component, but there's something that is likely coming from the nerve that is making only these muscle fibers, the strongest 2x2B muscle fibers, be the ones that are susceptible to aging effects. So if you now look at transdiaphragmatic pressure, the high amplitude, you see that all in the gray compared to the open figures, there's really no impaired ability to generate any of these levels of force. There's only impaired ability to generate the maximum force. These are, 75% survival, 2-year-old rats. They're old, but not terribly old. If you were to put that in human years, by the survivorship, we're talking somewhere in the 60 to 69. But it's not old. It's young. Right? So just to say that that already, you can already see that there is. So we also see then, if we label the entire phrenic motor neuron pool retrogradely, this is Matt Fogarty's work, postdoc in our lab, you can see that in the old rats, there's a reduced number of phrenic motor neurons. So again, the beauty of studying the system is it allows us to very carefully dissect all of the components and the contributions of these different things. And it's not, even though it's respiratory, and I believe that respiration is different than locomotion, it is. It still helps us understand what can be happening in other motor systems. So phrenic motor neurons are reduced by around 20%. Maximal transdiaphragmatic pressure reduced by about 25%. And the loss, again, is this is just a histogram now. This is the number of phrenic motor neurons on the y-axis, the size of the motor neuron on the x-axis. I mentioned that the size is a reflection of motor unit type. The units that are lost, only seen in the young animals, the open bars, are the largest units, right? The motor neurons that are remaining, they didn't all shrink. It's just you lost the really big ones. So you're using 20% of them, but you're losing the strongest ones. Again, rendering all of us susceptible to those respiratory complications. What happens to the neuromuscular transmission in old age? So these are now mice. With age, neuromuscular transmission failure is worsened already by 18 months of age. That's 50% survival. That's 90% survival. So really, this is closer to the 55 to 59. Notice that the bins are not the same size. But by that time, you already see a deficit in neuromuscular transmission failure that is sustained by 24 months of age. But if we give brain-derived neurotrophic factor, we saw that in the young animals, there's an improvement in neuromuscular transmission failure. Look at this. At 18 months of age, you can make them way better. Similar effects than you had at 6 months of age. So even though their baseline, without BDNF treatment condition, is one where there is impairment in neuromuscular transmission, you can still restore it. Now, by 24 months of age, you no longer can do anything about it with BDNF. Right? So this prompted us to do, I'm going to tell you, just a failed experiment where we used orally available phytoestrogen derivative that is a BDNF analog. It's called 7-H-dihydroflavone. Maybe some of you have heard about flavonoids and maybe use them in your diets. I don't know, but there are estrogen derived from plants that has effects similar to BDNF. So we gave dozens of mice that at age 18, waiting to see what happened at 24 months of age, and Sarah published a largely negative result. So we were not able to change the course. So there's something else driving this, not lack of BDNF availability, right? So it's positive. We learned that it's not the lack of the ligand, it's probably something with the receptor. So our ongoing studies are looking at what is driving the changes in TrkB receptor expression. Because here TrkB is still available, but here it probably is no longer available, right? And that's one of the things that happen. And just having more BDNF doesn't keep the TrkB receptor around, which was what we were thinking initially, right? So again, that was a smaller step. It didn't have the splash. If we had had success, it would have been, you would know of it by now, I know, I'm sure. But since it didn't, we still published it so that we understand. So if we give one an MPP1, the TrkB kinase activity inhibitor in these mice that are normal until we give them one an MPP1, you can see that the effects are the same. So what we think is BDNF and TrkB really set the min and the max for what happens in neuromuscular transmission, regardless of age group. Right? So again, finding a common mechanism of neuromuscular, even in old. So what happens to lipid RAF trafficking? Again, Maria and Matt now did some of this work. At low levels of activation, there's no differences between age groups in the type 1 or 2A fibers. But at the higher levels of activation, there is a difference, but only for the 2S and 2B motor units. So the muscle fibers that are innervated by the fatigable motor units have disrupted lipid RAF trafficking in old age. And it's only at the high stimulation frequencies. Again, what we're thinking is that trophic interaction is disrupted because lipid RAF trafficking is disrupted. So some of our nanoparticle work now we're using to use that, or using the nanoparticles as a way of understanding lipid RAF trafficking, because they get taken up by lipid RAFs, and studying other common mechanisms of disease, such as autophagy. We have several studies looking at that. So, that from this short part of the story, I've shown you that neuromuscular transmission is regulated by neurotrophins, and by TrkB receptor activation throughout the lifespan, that TrkB kinase activity is necessary for neuromuscular transmission, and that disrupting this can contribute to age-related impairment in respiratory muscles, but we think likely other muscles. So what happens in people? Right, so I've talked to you about mice and rats, some humans here and there, but in people, if you look at someone that has the same chronological age, or a group of people of the same chronological age, you all know that we don't look the same. We look very different. But the mice and the rats that we use have all been inbred. Even the outbred species are inbred compared to humans, right? So we're much more variable. So one of the things in which we can characterize human aging is by using something like frailty. So frailty is a syndrome that is characterized by declines in the physiological function of multiple organ systems. We think frailty is a major public health burden, and frailty and arthritis are very closely intertwined. So with early career faculty, Becky Johnson, we did some studies looking at frailty in patients that come for surgery because of arthritis. So we used knee or hip arthroplasty surgeries. They're elective surgeries. They're done mostly for pain, for people with pain from arthritis, and looked at their frailty indices. In the X, you have the bins of the different age groups, from 50 to 90 plus. On the Y, you have the frailty index. So frailty goes up with age, but the variability across each age group vastly overwhelms whatever age-related effects, right? So you can see some really young folks that are really frail, and some really not so young folks that are not frail at all, right? So in people, we can't use years anymore, right? So we're thinking we need to use frailty. Why? Because if we use frailty, this is mortality. So on the x-axis, you have months after the total hip arthroplasty. On the y-axis, you have incidence of mortality. In blue, you have the non-frail folks. It's not that they don't die, they die but at very lower rates. And in red, you have the frail folks, regardless of age. Right, so if you do multiple regression, then you consider age as a factor, is still I see these are just. So what about other things that we do to the respiratory system? So if you look at sedation and anesthesia, and you look at the number of events for breathing rate, and an awake animal, they breathe really fast. In an anesthetized animal, they don't breathe quite as fast. If you expose them to 10%, 5% CO2, breathing becomes a bit more regular, regardless of whether they're awake or anesthetized. But this is what I want all of us to remember, is that when you have light levels of anesthesia, even those that don't really impair the animal's ability to breathe regularly, you still impair the ability to generate some of the higher level forces. So the size that are not the highest, but somewhere in between, are already impaired with light levels of anesthesia. So we're thinking that this can be a test for us to see how frail you are. You give people a very mild sedative, something that we would do routinely for many diagnostic tests, and then we could measure EMG activity, and if their inability to generate size or respond to airway occlusion is impaired, that would suggest that their reserve capacity is impaired, right? Regardless of their age, based on their fit. So this is my last slide. So I hope I've shown you that sarcopenia and neuromuscular transmission failure decrease that reserve capacity, and that predisposes aged individuals to respiratory complications and death. And I'm going to leave it there.
Questions?
Yes.
The recovery experience was interesting, where you had the spinal cord injury and tried to induce the TREC-B expression 3 days post-injury, right?
Yes.
You were unable to recover that, basically, at least sufficient.
Yes. So our recovery was not as large as if the animals had constitutively higher levels or lower levels.
It was before the. So do you have anything what's going on? Is that just purely death of the motor neuron?
So the model, fortunately, the model that we use is a hemisection at a higher level than where the motor neurons are. So motor neuron death is unlikely. We haven't looked for it since the injury is not at the level of the motor neurons. But we actually think it just takes a while for the TREC-B receptor to become expressed after viral transfection, usually in the order of 2 to 3 weeks with AAV constructs. And we think that there is a window where the BDNF and TREC-B are most important. And there's a window early on where that plasticity can be enhanced. And if you get at it a little too late, then you can't really recapitulate that. But it's an ongoing area for investigation. So again, that's where that genetic mouse model with the knock-in construct comes in for them taking it away. We've been trying to explore ways in which we can restore the function faster than by just increasing the expression of the receptor.
Actually, following up on that, when you have a spinal cord injury, the endogenous response also is there is a recovery, and there's a short period during which there is a change. There is a endogenous response to trying to improve the levels, right? So I wonder if that time lines up with what you're doing with the external delivery with the endogenous receptor response.
Yes, very good point. So regardless of the model, whether it's with treatment or so on, in our model of spinal cord injury, we don't see much more enhanced recovery beyond 3 to 4 weeks. So whatever level of recovery we got by then, we don't really see it much beyond that. Now, some groups have recently been publishing studies where they look at these animals way later and showing that they can restore some functions by using chondroites. It's a way of digesting the scar that forms around central nervous system neurons after injury. And they suggest that they can get levels way further into the future. The challenge is that they're not directly comparable. They use a different strain of rat, we use female rats, we use male rats, and the extent of recovery that we see compared to theirs is also different. As I mentioned earlier this morning, I just came from the International Society for Neural Regeneration. That was one of the sessions, was how do we try to harmonize, so to speak, all these varying results that people are getting. And it's not a replicability issue, it's just that we're using slightly different versions of it. So how do you recreate the elephant in the room by touching the different parts, right? Everyone's familiar with that type of experiment. How do we keep the picture consistent? So it could be that the timeline is defined by mechanisms of spontaneous recovery that we're just enhancing and having them come in early, but we're not really adding to what the body is able to do anyway. But after all, that's part of what we do with regeneration. We're not trying to do superhumans necessarily. We're trying to restore what they Yes.
If you're finding that it's the fatigable fibers that atrophy during aging, does that suggest that coughing and sighing should be part of the physical therapy battery for people getting older?
Yes, absolutely.
And that's something that you suppose you could strengthen those fibers, and it's not, for example, that you lose them and no matter what you do.
Yes, very, very good point. So, of course, all of the work that I was showing was to understand mechanism, right? So that we can link it with therapy. But if we jump to therapy, I think that muscle strengthening and training should be matched to the type of units that you're losing. So in the old age, you should strengthen and try to activate these by something that has that threshold for activation. So there's different levels of doing respiratory therapy. Anyone does respiratory therapy here? So that's what I thought. So you can do inspiratory resistive training where you have just a resistor. So it makes it difficult for every breath. But that really is mostly activating the fatigue resistant units. But you can do inspiratory threshold training or cough or other behaviors where you actually have to generate a certain level of force, and that's what you're targeting for. Because that's there are some groups that have been trying to use that in patients, including after spinal injury. In old age, I think it makes perfect sense. And we think that they develop pneumonia most of the time because they have impaired swallowing. That's what people think of, is because they don't swallow well. Actually, if they can't cough effectively, you know, that even the small levels, because all of us have deficits in our swallowing, normal, and you're always having microaspirations, right? So microaspirations are not totally abnormal. But when you have something more than just a microaspiration, you're coughing, it's done, right? You never develop anything. But if you can't cough effectively, now you can be at risk. That's our friends. I did see our hand raised. Who was it? Okay. Yeah.
So you're trying to quantify frailty with this new method that you're proposing. What's like the current parameters that go into that index?
Yeah. So there's various ways of quantifying frailty, right? So one of the ways is doing a very comprehensive battery of force measurements and strength measurements and things like that. Another one is a quantification, an aggregation of deficits method, or accumulation of deficits. So you just count how many physiological organ systems are abnormal in some way, and just quantify that. The one that we used with Dr. Johnson for the frailty index is an accumulation of deficits plus activities of daily living. So that's, again, but as you can see, chronological age gives you a really, really wide range. And I would think that a person that is frail, age still matters, right? And I think that our frailty indices are just imperfect. In anesthesia, when we're thinking of patients that are going to undergo surgery in anesthesia, we're thinking of a way of quantifying their ability to undergo surgery safely. One of the best predictors of how people will do is, can they go up 2 flights of stairs? So if I ask someone, can you go up 2 flights of stairs, not stopping, you know, maybe carrying some things, and they say, oh yes, my house has a basement and a thing, and I go up and down all day, I almost can stop and say, you're going to do just fine. What we do for you is going to be fine. If they have trouble, then that's a big thing. Other people use the timed up and go, right? So it's just how fast do you stand up from the chair, and how far can you go, and other measures like that. There are a bit integrated responses, but the current methods that's most widely used is the accumulation of deficits. And I think it's a very imperfect way. Because for you have severe cardiovascular disease or mild cardiovascular disease, that counts as a deficit, right? Either one. And obviously they're not the same. Good question. Other questions? If not, I look forward to meeting with the students soon, or any of you throughout the day. Thanks for your attention.